Welcome to the Explainer. Today, our goal is pretty simple, but honestly, it's incredibly ambitious. We're going to try to save lab directors a massive amount of time and budget. If you're in preclinical research, you already know that trial and error just eats up your resources faster than literally anything else. So today, we're building a video summary of the optimized AKI-MSC therapy blueprint. We're extracting a highly actionable, standardized blueprint for acute kidney injury, or AKI, straight from a really complex Prospero meta-analysis. We're cutting right through all the guesswork and breaking this crucial data down into a clear, step-by-step explainer. Okay, let's dive right into this. We are essentially taking this massive sea of global literature and synthesizing it into one standardized, highly optimized protocol for in vivo AKI mesenchymal stem cell, or MSC experiments. The data we're looking at perfectly captures what we're trying to do here, taking scattered, completely disconnected data points from labs all over the world and bringing them together into a single, cohesive framework for your preclinical experimental design. Think of this explainer as a journey to find the signal through the noise. We're going to cover the data chaos, filtering the noise, proving baseline efficacy, a pretty huge delivery route surprise, finding the optimal dose, and finally, the standardized protocol itself. Let's get right to it.
Section 1. The data chaos.
Now, to really appreciate the solution here, we first have to just acknowledge the messy reality of scientific research. It can be incredibly frustrating, right? If you decide to research MSC therapy for AKI, you are instantly hit with over 3,000 unstandardized publications. And honestly, the issue isn't even just the sheer volume of papers, it's the wild variations within them. You've got different labs using completely different animal models, wildly varying methodologies, and dosages that are just all over the map. When everyone is doing something completely different, replicating results is practically impossible. And you know what that leads to? Hugely expensive trial and error for any lab just trying to establish a simple baseline.
Section 2. Filtering the noise.
So how in the world do we take over 3,000 chaotic papers and actually find the truth? Well, you have to ruthlessly filter the noise. The researchers here utilize the PRISMA methodology. This relies on super strict keyword and verifiability filtering to ensure linear, reproducible datasets. They took those 3,000 plus publications and aggressively narrowed them down to just 14 core verified papers. Those 14 papers, they form the absolute reliable foundation of this entire meta-analysis. And to account for the intra-study variance, because, even those 14 used distinct experimental materials, they utilized a random effects model as their statistical engine. By analyzing those 14 golden papers, we finally start to see some distinct, standardized parameters emerge for animal subjects. For the strain, Sprague-Dawley, or SD rats, are definitively the dominant industry standard. The data notes that while the Worcester strain is technically an alternative, they're highly aggressive, which makes SD the much preferred choice. For sex, there's a strong preference for males. And for physical specs, the ideal subject is 6 to 8 weeks old, so about 1.5 to 2 months, and standardized at over 200 grams in weight. Boom! This gives you a crystal clear baseline for your cohorts before you even touch a pipette.
Section 3. Proving baseline efficacy. Now that we've got the subject parameters locked down, let's shift gears and look at the actual results of the therapy. Does it actually work? And this brilliantly illustrates that it does. The overarching meta-analysis definitively confirms a negative 5.6 net reduction in serum creatinine when you compare the MSC experimental group to the control group. Now, since serum creatinine is a primary AKI biomarker, seeing a definitive drop exactly like this proves that the MSC intervention is effective. Period. Efficacy is just no longer a question we need to answer. From here on out, the goal is entirely about maximizing it.
Section 4. The delivery route surprise.
So maximizing that efficacy brings us to our first major pivot point in the data. And honestly, it's a pretty huge surprise. It completely challenges a very common scientific assumption. A lot of researchers logically assume that if you have a localized kidney injury, a direct localized injection right into the kidney is naturally going to be vastly superior to just injecting the cells into the overall system. Makes sense, right? But what does the data actually say? Well, now, what's really interesting about this is that the data profoundly shatters that localized assumption. Just look at the numbers. When controlling the variable at a 100 microgram MSC dose, the local delivery, meaning the subcapsular direct renal injection, only yielded a serum creatinine reduction of negative 0.79. But the systemic delivery, administered via tail vein IV injection, that yielded a massive negative 5.51 reduction. That is a staggering delta of 4.72. Counterintuitively, systemic tail vein administration drastically outperforms localized injection. No question.
Section 5. Finding the optimal dose.
All right, with the delivery route settled, we move to the final crucial variable that needs optimization before our protocol can be totally established: the dosage. And here we face another really counterintuitive revelation. The logical assumption in a lot of therapies is simply more is better. If a small dose works well, a massive dose should work wonders, right? Well, let's look at the data reality.
The data clearly identifies a therapeutic sweet spot, and it is actually not the highest dose. Take a look at the optimal dose of 100 micrograms. It gives us an apoptosis reduction of -5.34. That is a massive 3.12 improvement over the low dose of 30 micrograms. But look what happens when you push the dosage to the high dose of 250 micrograms. The serum creatinine and injury score reduction literally drops to -2.07. That's a 0.93 drop in efficacy compared to the 100 microgram dose. So pushing the dosage higher actually decreases efficacy, making 100 micrograms the absolute, undeniable therapeutic sweet spot.
Section 6. The standardized protocol. Okay, we've filtered the noise, proven the efficacy, discovered the best delivery route, and locked in the absolute optimal dosage. It's time to bring all this refined data together to complete our video summary. So, the crucial point is this. Here is your final standardized 4-step blueprint. Step 1. Use male Sprague Dolly rats 6 to 8 weeks old weighing over 200 grams. Step 2. Use standardized surgical nephrectomy or chemical cisplatin for the induction method. Step 3. Strictly prioritize systemic tail vein IV injection over local delivery. And Step 4. Utilize a target dosage of exactly 100 micrograms to maximize both serum creatinine and apoptosis reduction. Lab directors can basically take this blueprint, immediately implement it, and completely bypass that whole messy trial and error phase. It ensures maximum efficacy and saves a ton of resources. We've seen how stripping away the noise of over 3,000 papers can leave us with a crystal clear, highly effective methodology for acute kidney injury. But honestly, it leaves us with a much bigger question to think about. How could applying this exact same rigorous meta-analysis filtering accelerate your other regenerative medicine pipelines? I mean, what other massive breakthroughs are just hiding right now in plain sight, buried under the chaos of unstandardized data? Thank you so much for joining me on this explainer. See you next time.